Sometimes science plays out just like a great detective story. You've got a victim, you've got a weapon, and a whole lineup of suspects. But in this case, the crime scene is at the molecular level. We're on the hunt for a killer compound, and it's hiding inside one of nature's absolute most potent poisons. So let's get this investigation started. So here's the central mystery, right? We know which animal is responsible, but we don't know exactly how its weapon works. What's the secret sauce? What is the precise mechanism that makes it so incredibly fatal and so terrifyingly fast? So meet our prime suspect. This is the Australian box jellyfish, Chironex fleckeri. And this isn't just any jellyfish. It's widely considered the most venomous animal on the entire planet. And trust me, its reputation is well-earned and absolutely chilling.
And here is exactly why. Its venom can cause a human to die from cardiovascular failure in just a few minutes. That's speed. That's the most terrifying part of its whole profile. There's almost no time to do anything. But how? How does it pull this off? What is the secret to its lethal efficiency?
Well, this brings us right to the heart of our investigation. Is the venom's power all down to one single super potent killer molecule? Or is something way more complex going on here? Could it be a conspiracy? A whole team of toxins working together to create such a devastating result? That is exactly what a team of scientists set out to discover.
Okay, so the first step in any investigation, right? You've got to identify all the potential culprits. For these scientists, that meant deconstructing the weapon itself, breaking down the complex venom to see exactly what it was made of.
To do this, they use this really powerful tool called an FPLC. The easiest way to think about it is as a high-tech molecular sorting machine. Its entire job is to take a complex chemical soup, like venom, and carefully separate it into all of its individual ingredients. So here's how this sorting machine actually works. First, they inject the raw venom into this long column. Then, a solution pushes it all through. And it's kind of like a race. The larger, heavier molecules, they move faster and they come out first, while the smaller ones kind of lag behind. The machine then collects these groups as they exit, giving the scientists these distinct, isolated parts of the original venom. And boom, here's the result. This graph shows the venom's profile after it's been sorted. Each one of those peaks represents a different group of molecules, all separated out by size. So the investigators divided them into 6 distinct groups, or fractions. And this is our lineup. Meet the 6 main suspects in the death of a heart cell.
All right, now that the suspects were all isolated, it was time for the next phase, a direct confrontation. The plan was to expose the victim, living heart cells, to each suspect one by one to see which one was the real killer. The setup for the experiment was actually pretty simple. On one side, you have the suspects, each of the 6 venom fractions, a version where they remixed all 6 back together, and, of course, the original, untouched venom. And on the other side, the victim, a culture of living human heart cells.
So how do you watch this showdown happen in real time? Well, they used a system called xCELLigence. Just think of it like a life support monitor, but for cells. It measures how well the cells are sticking to the bottom of the plate. When a cell dies, it detaches. So by measuring this attachment, the scientists could get a precise, minute-by-minute reading of when and how fast the cells were dying. Pretty cool, huh? So the stage was set. The suspects were lined up, the victim was ready, the monitor was running. It was time for the verdict.
Now, take a look at this graph. See those lines staying flat at the top? For 5 of our 6 suspects, that's fractions 1, 3, 4, 5, and 6, nothing happens. The heart cells just stay perfectly attached, perfectly healthy. Turns out, 5 of our suspects were completely innocent. They were harmless. But then, look at the line for fraction number 2. See how it starts to drop? That means the cells are dying. So we have our culprit. Suspect number 2 is toxic. But wait a second. Look at the timeline. It's taking about 30 minutes to kill most of the cells. So yeah, it's a killer, but it's not nearly fast enough to explain the original venom's terrifying speed. And then came the final test. The original, untouched venom. And this is where it gets crazy. Look at that bottom line. It doesn't just drop, it plummets. The monitor basically flatlined, 100% cell death in under 10 minutes. So, while fraction 2 is the killer, it's nowhere near as deadly on its own as it is inside the original venom.
Okay, so let's just quickly summarize the evidence. 5 of the fractions, totally harmless. Fraction 2 on its own, toxic but kind of slow.
When the scientists remixed all the fractions back together, it was more effective but still not as fast as the original. And the original, whole venom, it was hypertoxic. So the mystery wasn't solved, it had actually just gotten deeper.
All of this evidence pointed the investigation toward a final and, honestly, really elegant conclusion. It wasn't about a single killer, after all. It was about the conspiracy. It was about the power of synergy.
Synergy is this amazing concept where the total effect of a team is way, way greater than what each member could do on their own. It's the classic 1 + 1 = 3 idea. The different parts work together to amplify each other's power.
And this chart just illustrates that concept perfectly. On its own, the killer, fraction 2, is slow. When you put it back with its team, the recombined fractions, it gets faster. But in its original, perfectly balanced environment, the whole venom, its speed is just off the charts. Those supposedly harmless fractions, they aren't useless at all. They're acting as accomplices, making the main killer brutally efficient.
And look, you don't just have to take my word for it. Here it is in the scientists' own words. They concluded that, yes, there is one main component that causes the heart cells to die, but it acts synergistically with the other parts of the venom to get the job done. Turns out, the killer needs its crew.
So the case is closed. The box jellyfish's secret isn't some single silver bullet, but a complex chemical cocktail, perfected by evolution. And that leaves us with one last, really fascinating question to think about. How many other poisons out there in nature, from snakes to spiders to plants, don't rely on a lone assassin, but on a deadly, synergistic conspiracy?